In fact, I want to start by saying thank you. Thank you to a big group of international current and former lab members, to colleagues, collaborators, mentors, and first and foremost today to the Australian National Academy for the big honor of having me elected as a corresponding member. As you can see from this slide, quite symbolically, there is a lot I can learn from coming to Australia. This is a symbol. It was actually taken during a retreat that we had with collaborators here from ANU, Thomas Price's group, and a lot of very interesting and exciting scientific interchange. And I hope that my having become a corresponding member of this academy will help contribute more to bringing to benefiting from the science here and bringing some of ours. If you think about what we have done scientifically, it's really one of the first lessons is how intuition can fool you. So if you take the question of where is the sun, intuition tells you, well, it's right there. It's in the sky, high and bright. But we all know that there are many more other suns out there in our Milky Way and other galaxies. And this is something that intuition initially hides from us. So the first contribution, I think, that we made to science was if one addresses the question of where do genes get regulated inside of human and other cells. And the intuitive answer that molecular biologists would give you, it's inside of the nucleus. That's where the DNA is, our hereditary material. That's where RNA is being transcribed and then exported as a messenger RNA. You can see it here. And then what happens in the so-called cytoplasm is that the protein biosynthetic machinery gets on and makes proteins. But the regulation by transcription, by epigenetic regulation, by RNA processing happens in the nucleus. Well, what we realized many, many years ago is that there is gene regulation in the cytoplasm, and that this process that is called translation, as well as the stability, the survival of messenger RNA, can be regulated. So how did we come upon this? When I was a postdoc in the lab of Rick Klausner at NIH, I was involved in the discovery of this very small RNA molecule called IRE for iron responsive element. It's made of RNA, not of DNA. And in fact, it was the first element to be found in mature messenger RNA to do an important job. And that job is to bind a particular protein, an RNA-binding protein called IRP1 for iron regulatory protein 1. And when this protein binds to this element, very interesting things happen. When it binds to the head end of the messenger RNAs encoding ferritin, H chain and light chain, the binding of this protein blocks the synthesis of ferritin protein. However, if it binds to the tail end of the transferrin receptor messenger RNA, it stops this very hungry nuclease that wants to eat up this messenger RNA from having its lovely meal. And so it stabilizes the RNA, allows it to survive, and it can give rise to more protein. And in fact, using this information and this setting, what we learned was the very first example of how translation can be controlled by an RNA element and a regulatory RNA binding protein. So here you see the process of translation being initiated. This is a bunch of helpers that all have their own names. This is the small subunit of the machine that makes proteins called the ribosome. And so this is how normally translation is initiated. However, when this protein IRP1 binds, the helpers are trying to get the ribosome to do its job, but IRP1 sort of sits like a truck on a landing pad and prevents this from happening. In fact, this doesn't only happen on ferritin messenger RNA, but we realized a bit after that IRP serves almost like a conductor of an orchestra. So in addition to regulating ferritin, it regulates a number of other very important messenger RNAs that encode proteins that all have some importance in iron metabolism, and in fact there are some human diseases associated with this. It also regulates the stability of another messenger RNA that we know of that is important for iron uptake. So it has this conductor function of coordinating a network of messenger RNAs and regulating its expression. So, if you like, research that we did and that others did around the world led to the realization that in addition to gene regulation in the nucleus, gene regulation in the cytoplasm is very common. So it's a little bit like the sun and the galaxies. So what I'd like to cover with the rest of my time is what we are currently doing. And you will see how our current research very much relates to these now quite traditional discoveries. What we try to understand is the question of how our genetic makeup, our DNA, if you want, often referred to as nature, and our lifestyles come together, communicate with each other, and make us into what we are, whether we are healthy, unhealthy, etc. And so the question of how the regulation of our genes and the metabolism that is brought about by our lifestyles, how they talk to each other. There's a lot that remains to be learned, and we're trying to uncover some of those secrets. And so let us return back to this old friend, IRP1, which I already introduced to you as an RNA-binding protein. And I only told you half of the truth, because IRP1 is only an RNA-binding protein when within cells the iron levels are low. Let's watch and see what happens when iron levels rise. You can see that I arrested it slightly too early, so you only faintly see it in the background. What you can see is that there is this cube that moved in. This cube is made of iron and sulfur. And when this cube comes into the protein, it changes its shape. It can no longer bind to this iron-responsive element. That actually tells you how iron regulates this protein. But what has now happened is that this protein that used to be a genetic regulatory protein has turned into an enzyme. And you can see it faintly. It's cytosolic aconitase. It's doing a completely different job. It now plays a role in metabolism. And if I already, when I practiced yesterday, had a problem with this, so I now let it run one more time. So you can see it turns into aconitase, and when iron levels get low again, it's a reversible phenomenon. So one protein can do two very different jobs. So on the basis of this observation, Thomas Preiss here at ANU and I hypothesized a few years ago that maybe that could harbor some of the secret of how metabolism and gene regulation could talk to each other. Could there be multiple such double job proteins that play a role in metabolism as well as in gene regulation? And so in order to find out, we developed a brand new technique. It's called RNA interactome capture. It's explained on this slide. But what in essence it does, it can capture all the proteins that are active in binding RNA in a living cell. And through this, we realized that the universe of RNA binding proteins is much bigger than had previously been anticipated. And most importantly, that it includes about 60 or 70 of proteins that were known to play roles in metabolism as enzymes, and that we now found also have the ability to bind RNA, just like this IRP1 aconitase example I gave to you before. So what you see behind me is one of the central pathways of metabolism that is existing in all cells. It's called glycolysis. It's the pathway through which glucose, blood sugar, is being broken down into fuels and into building blocks for cells. And when you see a green dot in one of the several enzymes that are leading through this pathway, it indicates that this metabolic enzyme can also bind RNA. And you see a lot of green dots behind me. When they're on the left, they tell you that the enzyme in the brewer's yeast, Saccharomyces cerevisiae, is also an RNA-binding protein. When it's on the right, it says that the human liver protein is doing this, and you see a lot of green proteins across. So nature, evolution, has conserved the ability of these bifunctional proteins, and we think that there is a lot of secrets that we will be able to unlock in the future by understanding what this dual role may mean. So to summarize in rather simple terms, what we are realizing is that this class of new proteins, we call them enigma-RBPs, because their RNA binding activity is enigmatic, while we know very well what their enzymatic function is. That these enigma-RBPs are not like cameras or home phones or record players fulfilling one particular role. But what they seem to be is smartphone proteins that can integrate multiple functions in gene regulation and in metabolism and serve as a communication tool between these two worlds. So with this, I'm very happy to realize that I'm done also ahead of time. I was so scared I might not make it. That I have plenty of time to thank the Academy once again, and to also express how much I look forward of interacting with the Australian scientific community even more than I have in the past. Thank you very much.
Thank you, Matthias, again being ahead of time. Are there any questions or comments? Please, Ian.
Thank you, Matthias. Wonderful talk. Is there a possibility now of doing one of the other type of capture experiments where you actually capture the small ligand that's binding to the RNA so that you can actually, I think there's experiments where this has been done for, say, molecules that bind to steroids in cells, or sterols, sorry, in cells.
Okay, what you are asking, if I understood your question correctly, is I have mostly spoken about the protein and about the RNA, but there was also on one of the slides that showed the triangle, metabolites and small molecules that would affect this, just like iron affects IRP1 and its functions. So I think that within the model that I presented, small molecules will play a role in some such REM networks, as we call them. And indeed, there are techniques, mass spec-based and others, that should be able to identify these. But we also think that there is another possibility of how this regulation could happen. And that is that some of the metabolic intermediates of our daily metabolism, such as acetyl-CoA, for example, is able to modify proteins in a way that depends on the level of acetyl-CoA by acetylation, as you know very well. And so we think that reversible modifications of proteins that are driven by metabolism could influence these RNA-protein interactions as well. And again, be a tool to foster the communication between gene regulation and metabolism. Thank you for the question.
Any further questions? If not, could you please? Okay, please go ahead. Yeah, last question.
Give us an example of how those metabolic enzymes help to regulate the system when they act as an RNA binding. Are there any examples where it's known how that works?
Yeah, so in the case of IRP1 aconitase, I tried to illustrate how when it works as an RNA binding protein, how it orchestrates the expression of proteins that are important for iron metabolism. Another very interesting example is a glycolytic enzyme called GAPDH. So the GAPDH protein is very well known to be important for the glycolytic pathway. And it was discovered just about 3 years ago by the group of Erika Pierce that this protein can also bind to the messenger RNA that encodes a very important immune response molecule, gamma interferon. And so in T cells, cells of the immune system that help to fight cancer cells and various pathogens. In resting T cells, this gamma interferon isn't needed. And so the GAPDH protein, the enzyme, sits on the RNA and again prevents the protein synthesis machinery from making this. But when there is a pathogen or the T cells are activated, it dislodges from this. The cells change their metabolism totally, and now the gamma interferon protein is being made and secreted and sort of helps the immune job. And so these are two examples. The number of examples we really know of well is still limited, but as I mentioned, having discovered that some 60 or so well-known enzymes have these second jobs, there's a lot ahead of us for discovery.
On that note, could you please join me to thank Professor Matthias Hentze?